Okay, let's start.
Can we start, Dr. Senator?
Yes.
Dr. Nafise, can we start?
Hi, yes, we can start.
Yeah. Welcoming to the next talk. Let me introduce our friend, our moderator today. In fact, the two speakers for the day, next talk. Let me introduce Dr. Noroozi first. Dr. Noroozi started veterinary school in 2014 and graduated with honors in 2021 and got her Doctor of Veterinary Medicine degree. She has been working as a research assistant in the Experimental Research Center at Tehran University of Medical Sciences for almost 5 years. During this time, she has published 2 scientific articles in peer-reviewed journals and has 3 presentations at different conferences. Her scientific interests are memory impairment, infectious diseases, and metabolic disorders. So we're coming to next speaker, Maryam Shayan. She is currently working as a postdoctoral research fellow in the Department of Pharmacology at Tehran University of Medical Sciences. She is a physician scientist interested in neuroscience and neuro-ophthalmology. And their talk would be evaluation of the effect of polar and non-polar extracts of Symphytum kurdicum plant in preventing hemoglobin glycation in an in vitro condition. Over to you, Nafise.
Thank you so much for your kind introduction.
Hello, I'm Maryam Shayan. I am here with Nafise Noroozi to talk about the effect of Symphytum kurdicum extract on prevention of hemoglobin glycation in an in vitro model. First, we talk about diabetes disease. Diabetes is a chronic disease that occurs when the body cannot produce enough insulin or cannot use insulin effectively. 285 million people have had diabetes until 2010. By 2030, this will rise to 440 million worldwide. Every 6 seconds, a person dies from diabetes. Diabetes is a direct cause of 1.5 million deaths in 2019. Diabetes is a direct cause of 1.5 million deaths in 2019. And it will at least cost $327 billion in health expenditure in 2017. The prevalence of diabetes in Iran was about 7 million people in 2013. The current treatments that are available for diabetes are sulfonylurea, metformin, and thiazolide indiones. The side effects of these drugs consist of hyponatremia and hypoglycemia for sulfonylurea. Metformin causes lactic acidosis, and thiazolide indione causes weight gain and hypoglycemia. The other side effects for these drugs consist of non-specific symptoms like nausea, vomiting, diarrhea, epigastric pain, anorexia, tachypnea, and lethargy. Due to various side effects of these chemical treatments, traditional medicine started to use plants as a treatment for diabetes. Traditional medicine is all the knowledge and practices used in diagnosis, prevention, and elimination of the disease from ancient times to present. In Iran, traditional Iranian medicine started with Muhammad ibn Zakaria Razi, Ibn Sina, and Ismail Jorjani. They are the pioneers of traditional Iranian medicines. The advantages of this traditional medicine are convenient access, cost effectiveness, and fewer side effects. One of the drugs that we suggest can be used as a treatment for diabetes is Symphytum kurdicum. Symphytum kurdicum is a plant grown in April and May in north parts of Iran. It is from the family of Boraginaceae, and the genus is Symphytum and the species is Symphytum kurdicum. Here we investigated the effect of Symphytum kurdicum, the different extract of Symphytum kurdicum in an in vitro model of diabetes. Nafise will tell you more about the results.
Okay, we extracted the hemoglobin from healthy people, and we measured its concentration using the Bradford's method. After the hemoglobin was extracted, we incubated the hemoglobin in the presence and absence of Symphytum kurdicum plant and glucose in different concentration of glucose. The protective effects of Symphytum kurdicum extract against hemoglobin glycation was then evaluated through hemoglobin A1C using the iChroma device and immunofluorescence assay. Here is the iChroma device and our findings showed that glucose causes A1C hemoglobin glycation and changes its structure. Aqueous extract at doses of 5 and 10 milligram per kg and ethyl acetate extract at the doses of 5, 10, and 25 milligram per kg revealed significant protection against protein glycation in an in vitro model. Here you see our results. The aqueous extract and the methylmalonic extract of Symphytum kurdicum showed significant results. Here, the red column is a model of diabetic person that we made in vitro condition. And here you can see the effect of Symphytum kurdicum on other kinds of extracts, such as ethyl acetate and N-hexane extract, which showed good results, but not as good as aqueous or ethyl acetate or methanolic extract. Here you can see aspirin. We used aspirin as a positive control group. And you can see that aqueous extract and ethyl acetate extract showed much better results than aspirin. And as previously stated, all of the different doses of this plant, polar and non-polar extract of Symphytum kurdicum, were remarkably effective in lowering the A1C hemoglobin levels that are probably due to the anti-inflammatory and antioxidative effects of Symphytum kurdicum. Here, of course, our work is much bigger than this, but we try to talk about it in just 15 minutes. It shows the importance of traditional medicine and the effectiveness of using Symphytum kurdicum extract as an alternative medicine to Western medicine, and the less side effect, for example, no hepatotoxicity or nephrotoxicity. Thank you everyone for listening.
Thank you.